हेलो एवरीवन एंड वेलकम बैक टू द चैनल। इस वीडियो पे भी हम लोग नेक्स्ट 25 क्वेश्चन डिस्कस करेंगे ऑन द सेम टॉपिक माइक्रोबायोलॉजी। सो वेरी फर्स्ट क्वेश्चन, इट इज क्वेश्चन नंबर 51। द मेन सेल वॉल कंपोनेंट ऑफ बैक्टीरिया इज ऑप्शंस आर पेप्टाइडोलिपिड्स, cellulose, peptidoglycan, chitin. So here the right answer is peptidoglycan. It is the main component of cell wall of bacteria. The very next question. NAM and NAG units of peptidoglycans are arranged alternating to each other by dash bonds. तो हम सबको पता है कि NAM and NAG ये molecules one after another is connected by a glycosidic linkage, and this glycosidic linkage has a specific name. यहाँ पे कौन सा नाम appropriate है? So options are alpha 14, beta 14, alpha 16 or beta 16. So here the right answer is beta 14. Beta 14 glycosidic linkage is found in between NAG and NAM molecule of peptidoglycan. Very next question. Amino acid side chain are found on peptidoglycan from dash molecules. So here the options are NAM, NAG, both known. So here the right answer is NAM. NAM से side chains develop होता है peptidoglycan. In side chains होता है generally of amino acids in peptidoglycan. So very next question. Teichoic acid is found in cell wall of dash bacteria. So options are gram positive, gram negative, both or none of the above. So here the right answer is gram positive bacteria. यहाँ पे teichoic acid मिलता है. तो teichoic acid is a characteristic feature of gram positive bacteria or gram positive bacterial cell wall. The next question. Penicillin is a bactericide antibiotic, but it is not effective to gram positive, gram negative, both or none. So penicillin एक एंटीबायोटिक है जो कि बैक्टीरिया को इफेक्ट करता है. इट किल्स बैक्टीरिया, बट इट इज नॉट सो मच इफेक्टिव टू विच बैक्टीरिया. सो हियर द राइट आंसर इज ग्राम नेगेटिव. क्यों? क्योंकि पेनिसिलिन जनरली इफेक्ट करता है ग्राम पॉजिटिव बैक्टीरिया का पेप्टाइडोग्लाइकन लिंकेज को ब्रेक करने में, and in gram negative bacteria, as this peptidoglycan is very less in quantity, so यहाँ पे peptidoglycan का कुछ खास डैमेज होता नहीं है, and gram negative bacteria पेनिसिलिन जैसे एंटीबायोटिक से ज़्यादा कुछ इफेक्टेड होते नहीं है. So that's why gram negative bacteria are more dangerous when we are using penicillin to kill them. They may not be killed by this particular antibiotic. So next question. Which of the following bacteria lack of cell wall? So यहाँ पे कौन सा ऑप्शन सही है? एस्चेरिचिया कोलाई, माइकोबैक्टीरियम ट्यूबरकुलोसिस, माइकोप्लाज्मा न्यूमोनी, स्टेफिलोकोकस ऑरियस. इनमें से कौन सा बैक्टीरिया जहाँ पे सेल वॉल नहीं होते? सो राइट आंसर इज माइकोप्लाज्मा न्यूमोनी. यहाँ पे सेल वॉल नहीं होते. तो वेरी नेक्स्ट क्वेश्चन. बैक्टीरियल और फंगल सेल विद ए पार्शियली रिमूव्ड सेल वॉल इज कॉल्ड प्रोटोप्लास्ट, ल्यूकोप्लास्ट, माइकोप्लाज्मा और स्पेरोप्लास्ट. तो बैक्टीरिया जहाँ पे जनरली और बाय नेचुरल दे डू नॉट हैव सेल वॉल, देन वी कैन यूज द वर्ड माइकोप्लाज्मा. बट जब आर्टिफिशियल इनका सेल वॉल को रिमूव किया जाता है, देन द टर्म्स इज कॉल्ड एज स्फेरोप्लास्ट. राइट आंसर इज ऑप्शन डी, स्फेरोप्लास्ट. What is the primary function of mesosome in bacterial cells? Options are protein synthesis, ATP production, and DNA replication, photosynthesis, or movement of the cell. So here the right answer is ATP productions and DNA replication. So that is the main function of mesosome in bacteria. The next question. Polybeta hydroxybutyrate, that is what in shortcut PHB, is present as a reserve material in which of the organism? Options are algae, bacteria, fungi, plants. So this PHB is actually bacterial reserve material. So the right answer is option B. Next question. Which enzyme is predominantly found inside carboxysomes? Options are nitrogenase, Rubisco, ATP synthase और DNA polymerase. Generally carboxysome एक सेल ऑर्गेनेल है बैक्टीरिया, कुछ पर्टिकुलर बैक्टीरिया फोटोसिंथेटिक बैक्टीरिया में मिलता है, एंड इनके अंदर एक्चुअली कौन सा ये एंजाइम्स अवेलेबल है गिवन आउट ऑफ दिस गिवन ऑप्शंस. तो हियर द राइट ऑप्शन इज रुबिस्को. रुबिस्को एंजाइम अवेलेबल होता है कार्बोक्सिजोम सेल ऑर्गेनेल्स या कार्बोक्सिजोम बॉडी के अंदर किसका? इन सम फोटोसिंथेटिक बैक्टीरिया. The next question. Which function do magnetosomes prefer in magnetactic bacteria? Options are ATP productions, oxygen respiration, navigations along Earth's magnetic field, and number D is nitrogen fixation. So here the right answer is navigation along Earth's magnetic field. So generally कुछ particular magnetite particle containing bacteria In shortcut, we can say there is an organelle called the magnetosomes. These magnetosomes are present in सब magnetotactic bacteria, जिनका काम होता है अर्थ मैग्नेटिक फील्ड्स को रिकॉग्नाइज करके मूवमेंट में पार्टिसिपेट करना. सो द ऑप्शन सी इज हियर राइट, नेविगेशन along Earth's magnetic field. Generally, this magnetosome has another function. And वो function क्या है ना, it useful to break down of hydrogen peroxides, or you can say removal of hydrogen peroxides, that you can say detoxifications from H2O2. So these are the two functions of magnetosomes. Next question, which of the following best describes an episome? इनमें से कौन सा ऑप्शन एपिसोम्स के लिए अप्रोप्रिएट है? ऑप्शन है a plasmid that can integrate into the host genome, a type of bacteriophage, a membrane bound organelle in bacteria, a protein responsible for bacterial motility. So here the right option is number A, a plasmid that can integrate into the host genome, that is what called as episome. The next question, what is the primary functions of bacterial pili? So options are locomotion, DNA replication, attachment to surface and conjugations, or number D, protein synthesis. So here the right answer is attachment to surface and conjugation. That is the main function of pili. Next question, which protein primarily makes up bacterial flagella? Options are tubulin, actin, flagellin, or myosin. So right answer is flagellin, the main protein component for flagella, bacterial flagella. Next question, which of the following bacteria reproduce by budding? Options are Escherichia coli, Colobacter, Bacillus subtilis, and Clostridium. So here the right answer is Colobacter. It has the capacity to reproduce by budding. The next question, which of the following is the correct sequence of events in binary fission in bacteria? तो ऑप्शन क्या क्या देखते हैं. नंबर 1, A, DNA replication, then septum formation, then cell elongation, then cell division. Okay. Number B, cell elongation, DNA replication, septum formation, cell division, DNA replication, cell elongation, septum formation, cell division. Number D is septum formation, DNA replication, cell elongation, and then maybe cell division. So here the right answer is number C. First DNA replication, then cell elongation, then septum formation, and finally it is cell division. These are the proper sequence for this what called as binary fission. Next question, which of the following best describes bacterial transformation? The options are transfer of DNA through a direct cell-to-cell contact, uptake of free DNA from environment, transfers of DNA using bacteriophage, exchange of genetic material between fungi. So for transformation कौन सा statement exact है? So here the option is uptake of free DNA from the environment. For the transformation, this particular sentence is appropriate. Next question, who discovered the phenomenon of transformation in bacteria? The options are Oswald Avery, Frederick Griffith, Alfred Heresy, and Louis Pasteur. So here the right answer is Frederick Griffith, who first time approached or discovered this phenomena, you can say, and that is called as transformation. The very next question, what is required for artificial transformation in bacteria? Options are DNA polymerase, competency factors, we can say calcium chlorides, or electroporation, or restriction enzymes. So the right option, right answer is calcium chloride or electroporation are actually techniques required for this artificial bacterial transformation. The very next question, in genetic engineering, which bacteria is commonly used for transformation experiments? Options are Bacillus subtilis, Streptococcus pneumoniae, Escherichia coli, and Clostridium botulinum. So here the right answer is Escherichia coli, which is very much useful for transformation experiments, mainly for genetic engineering. The next question, which of the following can be used to induce competence in bacteria for transformations? So इनमें से कौन सा ऑप्शन है जो कि competence factors को एक्टिव करने के लिए यूज होता है, and this competency actually very much important factor for transformation, bacterial transformation. So options are heat shock, UV radiation, antibiotics, or lysosomes. So here the right answer is heat shock. heat shock से क्या होता है, ट्रांसफॉर्मेशन प्रोसेस के लिए कॉम्पिटेंसी जनरेट होता है. सो दैट इज द राइट आंसर. नेक्स्ट क्वेश्चन, व्हाट इज बैक्टीरियल ट्रांसडक्शन? So which answer is actually appropriate for this transduction? Options are transpose of DNA through direct contact between bacteria, transfers of DNA via bacteriophages, uptake of free DNA from the environment, or option D, mutation in bacterial DNA. So here the right answer is the transfer of DNA via bacteriophages. Next question, which scientist or scientists discovered bacterial transduction? Options are Frederick Griffith, Lederberg and Gender, Watson and Crick, or number D, Avery, MacLeod and McCarthy. So the right answer is Lederberg and Gender. They discovered together this process of transduction in bacteria. Next question, what are the two main types of ट्रांसडक्शन? तो जनरल ट्रांसडक्शन का दो टाइप्स होता है, इनमें से कौन सा अप्रोप्रिएट से? लाइसोजेनिक एंड लाइटिक, generalized and specialized, transformation and conjugation, induced and spontaneous. So here the right answer is number B, generalized and specialized. My next question, which type of bacteriophage is involved in specialized transduction? Lytic phage, lysogenic phage, retrovirus, non-enveloped virus. So here the right answer is lysogenic or temperate phage are required for exact gene sequence transfer, or you can say specialized transduction in bacterial recombination. My next question. Okay, so यहाँ पे हमारा सीरीज कंप्लीट होता है of these 75 questions. In next video again we are going to discuss the next 25 question. So thank you. I hope this video will be helpful to you. Thank you again.